Hi everyone, Professor Golden again. Today I want to talk to you about our genetic material. Our genetic material is also known as DNA. Have any of you ever heard of Ancestry.com, 23andMe? We're all very interested to find out what makes us us. So today I want to show you a little experiment that we do in our Biology 10-11 classes where we extract DNA from fruit. So this is an experiment that you could do from home. It's very simple, all the ingredients you can find in your home. So first we're gonna look at some fruits. Today we're gonna be working with strawberries. We're also gonna be working with a banana. You need some Ziploc bags because we're going to crush them up. It's gonna be great with our mortar and pestle. You need some funnels. Any containers will do. You could do a very short glass, you could do a larger glass. Today we're using 250 milliliter beakers. You need isopropyl alcohol, 91%, 100% is great. I know they're in shortages right now, but don't worry, we will get those back one day. We need some salt, just some sodium chloride, regular table salt, and also we need dishwashing detergent. Dawn can work, any other brands is perfectly fine. You can do this at the dinner table, all right? And so I'm gonna show you step by step on how to extract the DNA. So first thing you're gonna do is you're gonna put your fruit inside the Ziploc bag, and then here comes the fun part. You start smashing. You wanna smash it till it's a very fine pulp so that it's easier to go through the funnel. Strawberries are fun. So next up, we're gonna make our DNA extraction buffer. First thing you do, we have to measure everything out. So here I have 15 grams of table salt, sodium chloride. You can just eyeball it if you're doing it at home. It doesn't have to be as precise as it is in the lab. So we got 15 grams of salt. We're gonna do 2 cups of water here. Again, you could just measure it with tools that you have at home. If you have a measuring cup, even better. So 2 cups of water. And then we're gonna mix in 50 milliliters of Dawn dish soap. So basically this is just making a very soapy, salty water. This is gonna break down the molecules in the strawberry and extract the DNA from it. So now we're gonna mix our DNA extraction buffer with our samples. So you need around 10 milliliters of our soapy mixture. I have this cute little graduated cylinder. Again, it doesn't have to be as precise if you're doing it at home. Just eyeball it, it will still work. All right, and we're gonna add it to our samples. So as you can see, they've already been mashed up. We mashed them up earlier. I used 2 strawberries and I used half a banana. So we pour the 10 milliliters into the bag, close it up, mash it up a little bit more. And we're gonna do the same thing to the banana. So now we've added our DNA extraction buffer to our samples. So now we're gonna filter it through. We're gonna try to get a little bit of the chunks out because you want it to be a smooth mixture once we add our isopropyl alcohol to it. So all I have, a filter, I have a small beaker, like I said, at home you can use a glass, and then we're just using coffee filters. You can also use a moist paper towel if you would like, but as long as it's a little shield so that we can just have the liquid and not all the chunks. And then you can use a spoon, you can use anything just to try to help it filter out a little bit faster. Just trying to get all the liquid and none of the chunks. Overall, you want to try to get at least 10 to 15 milliliters of liquid so that you can easily see once the DNA floats to the top of the mixture. All right, so now we've filtered our samples. Now we're gonna move them to a smaller beaker just so that we could see it better on the video. So we're gonna pour our strawberry mixture in here, and then we're gonna pour our banana. So as you can see, the 2 strawberries produce a lot more liquid than the banana. So we have to sit here and think as scientists, does this have to do with the chromosomes that are found in both of the fruits? Would it have been different if we would have used the whole banana? All right, so now we're gonna add our 91% isopropyl alcohol. We're gonna be adding 20 milliliters to our samples. So now we just have to wait just a couple of minutes. It doesn't take too long. And what we're waiting for is like a white fluffy cloud to come up to the top of the liquid. What's that white fluffy cloud? That's the DNA. So now we've waited about 2 to 3 minutes, not too much time. Do you see this little cloudy mixture on the top of the banana? This is its DNA. So if you successfully extracted DNA from from a banana, if you want to do it from the strawberry, look at this. Look at how congealed it is. But now you have broken down all the phospholipid bilayers, you've broken everything down. And now we have DNA. So that concludes our experiment for DNA extraction. It takes around 15 minutes. It's family fun. You could do this with a bunch of different fruits. We've never tried it with vegetables. If you try it with a vegetable, make sure to email us at sciences.nsula.edu. Tell us how it is. Send us some pictures, send us some videos. We would love to see them. I hope everyone has a great rest of their day and weekend, and fork 'em demons.